Hello everyone, welcome back to the Genomics Bootcamp and the Introduction to Genomics lecture series. This time we will be talking about selection signatures. Before we go on, we do the usual summary from the previous lectures. So we continue to talk about SNP markers, so they are still a thing, and our discussion will be centered on them. We follow their inheritance patterns from parents to offspring on the basis of chromosomes. And on these chromosomes, we also observe the so-called recombination events. Another important aspect we talked about on the previous lectures is the so-called linkage disequilibrium, or LD in short. And this LD characterizes the degree of relationship within these inherited chromosome segments. What I mean with that is that if two SNPs are closer to each other, they are more likely to be inherited together to the next generation or from parents to offspring. This means that we observe these segments on the genome that tend to stick together and be inherited as one group rather than on a SNP-by-SNP basis. And we also talked about the so-called genome-wide association studies, or GWAS, that identify the parts of the genome that influence some phenotypes. Now I bring up the GWAS here because, as you will see, the interpretation, or in a general look and feel of the selection signatures, is very reminiscent of that from GWAS. But while in GWAS, we are trying to figure out the region of the genome that influences certain phenotype, in selection signatures, we try to figure out why is certain part of the genome more important in certain populations. Now let's start with the definition of the selection signatures. Well, I picked this one from a paper that you see below, and it says that selection signatures are defined as regions of the genome that harbor functionally important sequence variants and therefore are or have been under either natural or artificial selection, leaving special patterns of DNA behind. So I really like this quote, or this definition, because it's just really nicely summarizes what selection signatures are. So to recap in bullet points, so there is certainly a selection acting on all populations, certainly natural selection acting on all populations, and in case of livestock, also there is a strong component of artificial selection. Certainly, if we look at the genome, we can find the influence of this selection on the genomic level. And this influence we detect based on the special patterns of nucleotide variation that we find on the genome. Now, if we go into details what selection signatures are, so these are basically parts of the genome that are very similar across the population. And this similarity is caused by important sequences, or otherwise known as genes, that pop up in an ancestral genome. Now, this important gene is visualized with this line here. And because this part of the genome, or this gene, is so important, it is also preserved in the present day population. But of course, not the entirety of the ancestral haplotypes are preserved, but just some parts of it. Now, the extent of these haplotypes that are detectable in the present-day population, they are influenced by multiple factors, such as recombination rate, mutation rate, population size, and the strength of the selection. Another reason why we don't see the entirety of the ancestor haplotype in the present-day population is, of course, that the recombination is acting with each generation, which means that we find segments of different length in the present-day population. But you see that this important gene is still here, and also the region tightly around it. Now, this is another visual example actually showing the same. So there is a population here, and there is this one person who has a very beneficial gene here, or a very beneficial variant. Because of this variant give this person a selective advantage. This means that with time, this gene is more widespread in the population. But if you notice on this picture, so not just the gene itself is widespread, but also the surrounding regions. So you see here, there is a specific pattern here. So this is a darker colored block also just nearby, and also here a bit further away. And this whole segment is actually inherited in the future populations. And this is the specific pattern that is then detectable. And this is how we find the selection signatures in the populations. Or in the literature, you also find the expression selective sweep. This whole scenario that I just described, so there is this important gene and which drags along the surrounding regions, is the so-called genetic hitchhiking effect. Now also with the selection signatures, there are multiple possibilities which are described in these three rows. So this is the row A, B, and C. The first one is very similar to the one that I already mentioned, which is the so-called hard or classic selective sweep, where we have a population and then there is a certain mutation that is very beneficial. And this one with time spreads in the entirety of the population. So this region becomes entirely fixed. The second option that is shown here is the so-called soft selective sweep due to standing genetic variation. Here there is a certain population, and then for certain traits there is a certain variation, which is at the beginning neutral. So these blue lines and these green lines, they are basically equal. But let's say the environment changes and this mutation suddenly becomes very beneficial. So of course, selection, either natural or artificial or both, pick up on this, and this mutation also spreads in the population. Again, you see a very similar pattern. So there is the gene of interest or the region of interest, but also the surrounding regions are dragged along, which create a certain recognizable and detectable pattern. Now, the selection signatures or selection sweeps are not necessarily absolute, meaning that they not necessarily spread in the whole population as shown here. Now, the reason for this could be, well, different. It could be that the selection pressure started to act just relatively recently. So the beneficial allele had not had time to spread in the entirety of the population yet, but it is on its way. Or it could be also that the selection pressure is not so detrimental that only individuals having this certain allele or certain variant survive. So it could be that it just, well, not widespread in the entirety of the population for this reason. The third option here is the selection sweep with the multiple origin, which is quite similar to the scenario B here, but in the scenario C, so we have a beneficial variation in the population that start to spread, but then there is another beneficial mutation that pops up. So these two start to spread, and both of them are more widespread in the final population here in some proportion. Again, they are not widespread in the whole population, but they just show up in certain individuals. But as you see, there are specific detectable patterns again. Here I would mention that also if you want to read more about this, of course, there is plenty of literature, but I found this paper from Saravanan and co-authors very helpful. Also, this figure is from that paper. So if you're interested, I definitely recommend you to read this paper to get more information on this. Well, for the detection of the selection signatures, there are plenty of methodologies, and I actually don't want to go into details for any of these. I would mention just this very neat graph, which actually shows two important things. One is that if you're interested in selection signatures, first you need to decide if you want to look within the population or between populations, because then you need to use entirely different methods. And the second thing, this figure shows that even within the intra or interpopulation methods, there is a huge possibility of choice. So you can go ahead and find out plenty of things about the populations of your interest. If you also saw some other videos from this channel, then you might even recognize some of these methods, because for some, there are also practical guides on this very channel. So for example, there is a lot about the linkage disequilibrium, for the intrapopulation statistics. And also we talked a lot about FSTs, including practical examples. If you're interested in, well, more examples or more practical demonstrations from the selection signatures from these methods, or maybe some other ones, be sure to let me know in the comments. Well, to summarize again a little bit, the selection signatures work on molecular marker data. That means that we could use microsatellite SNPs or genome sequence data to localize them on the genome. One very important aspect is that the genetic map should be available, because at the end, we want to localize these important segments or these specific patterns on the genome. And we cannot localize it without a knowledge of the genetic map. So we cannot do anything if you don't know where these specific markers are. Now, I mentioned the genome-wide association studies, or GWAS, at the very beginning, because as you will see, the visualization of the selection signatures, at least on the figures, looks similar to GWAS. But there is one huge difference. Actually, there are two huge differences. The first difference is that unlike GWAS, the selection signatures look for fixed parts of the genome. So they can detect regions that contain fixed alleles, which is problematic for the GWAS analysis. The other big difference to GWAS is that for the selection signatures, we actually don't need phenotypes. This is a huge advantage because nowadays there is plenty of genotypes available. But if you look for certain phenotypes, sometimes any phenotypes, sometimes some specific phenotypes, you might have a very hard time to find them. Maybe they are just hard to find, hard to get from the respective organizations, or they just don't exist at all. On the other hand, you could find loads and loads of genotype data from freely available resources. Also, some of these resources and free open access data was highlighted in a number of videos on this channel. So if you are looking for data, be sure to check out those videos for open access data for the species of your choice. One additional note here, what I mean by that the phenotypes can be sometimes useful. Actually, if you have some phenotypes, even if this phenotype is nothing else but the breed identity or some kind of grouping of the individuals, you might use this in your research questions or how you set up the experiment. But if you don't have any phenotypes, you don't need to worry, because you can still run the selection signature analysis. As I mentioned, the selection signatures can be detected by specialized methods, as you have seen in the figure before. These could be within or across populations, in individual genotypes or pooled DNA samples. The interpretation is quite similar to GWAS. Actually, there are some plots and there are some high or low values that produce some kind of a signal. Here is an example of such a graph. So just don't worry about the statistics right now, but you see that there is something going on in this region, and depending on the statistic, there is a signal that indicates that something going on in this particular region, and this something is most likely a selection signature or selective sweep. Here is again a series of figures that actually demonstrates what happens in the selection signatures. This first one is a quite clear scenario of a Manhattan plot with several signals, but you see that there is one signal that towers above all of the others. So we will concentrate on this region. If you look at the allele frequency spectrum, you will see something very particular. So this is this part of the genome. So these are the megabases, and then this is the allele frequency of one of the alleles. So this is SNP data, meaning that the SNPs are biallelic. So the frequency of one allele is, let's say, 0, then the frequency of other allele is 1. So basically, only one allele frequency is enough to express the situation on that particular locus. On a, quote, normal end quote parts of the genome, you see something like this or this. So you see that some of the alleles are fixed one way or the other way, but there is a lot of other alleles that are intermediate in frequency. But at the place of this highest signal, we see a big hole here, meaning that the SNPs in the region tend to be fixed for one allele or the other allele, but there are virtually no SNPs that have the intermediate frequency. This is because this entire region is basically homozygous due to the genetic hitchhiking effect, as we described before. So there is this gene in the middle, or almost at the middle, which actually drags along all the surrounding regions. Now, this is the third part of this figure series, and you see that there are several methods used to detect the same selection signature. And while these plots look somewhat different, all of them actually point into the same direction, so that there is something going on in this particular region of the genome. And this something is most likely a selection signature. Now, I just wanted to mention briefly here the runs of homozygosities. So for the runs of homozygosities, there will be a separate video on this channel. So be sure to check out also that. But to explain briefly for this video, what we see here. So basically these small stripes here, or these small colored segments, are homozygous regions on the genome. So there is a stretch of the genome that consists of entirely homozygous markers. Now you see that the distribution of these runs of homozygosities is fairly random at the genome, but there are parts of the genome that tend to be homozygous at the very same spot for the entirety of the population, or the, well, genotyped individuals anyway. So these are the individuals. Each line here is an individual. And you see that, for example, this part of the genome, it tends to contain a run of homozygosity for almost all individuals, meaning that it is homozygous for all individuals, which is actually, again, this detectable pattern that points towards selection signatures. Also, for example, here, there is a small region that tends to be homozygous for everyone. So you see here in some individuals, there is a larger segment that are homozygous, but others, they have just this tiny, tiny homozygous segment, but still having this pattern, which also points towards a selection signature or a selective sweep. If you want to find out more about this, so this is the source paper for this figure. And I also wanted to show you some differences across populations. So this paper here studies some sheep populations. Well, this is a little description here. Also, it mentions 6 sheep breeds, but I just cut out these 4 figures because just to demonstrate my point. So basically, this left side of the figure is something similar that we have seen before. And the dashed line here is the actual point of interest, or the middle of the region. Now you see that in some breeds where there is absolutely nothing, so there is a lot of SNPs here with intermediate frequency, which means that there is no fixation here or no selection signature, meaning that this region on the genome was not selected for. Obviously, the same genes are present in the same region because all of these 4 are 4 sheep breeds, so they have the very comparable genome, but there is no selection acting in this population. In this other population here, you see a huge gap here, or this hole, meaning that the SNPs are either fixed one way or the other way. And this is a clear indication of a selection signature, meaning that there is a gene probably in here somewhere, which is really important for this breed. And again, because this gene was selected for, it dragged along the surrounding region, which created this pattern. Now, these 2 other breeds here are kind of an intermediate case. you see some effect of the selection, especially here. So there is a, well, not quite as widespread, but there is some fixation here in this particular region. And also in here, there is maybe even a bit less than previously, but also you can detect some kind of a selection signature or selective sweep. With this, I just wanted also to demonstrate that if you look at multiple populations from the same species, you can find very different outcomes or very different results. Of course, depending on the various natural or artificial selection pressures acting on these populations. We are already at the summary for this selection signature story. So we have learned that the selection, either natural or artificial, leaves detectable signals on the genome. These signals usually center around the beneficial gene or genes, which drag the surrounding genomic regions with them. This so-called genetic hitchhiking effect is actually caused by linkage disequilibrium, meaning that the surrounding regions for any particular nucleotide tend to be inherited together. And if this nucleotide or this gene tends to be a very important one from the viewpoint of selection, then this spreads in the population and with it its surrounding regions as well. So we actually see these small parts of the genome that are very same in the entirety of the population. Of course, here are also various options. It could be a so-called hard sweep, which is in the entirety of the population, or a soft sweep, which is in part of the population. Also, there are various ways to detect selection signatures. There's plenty of methods, approaches, and software which look for, well, homozygous regions throughout the population or some kind of pattern that could be used to identify such particular patterns. Of course, depends very much if you are looking within population, which is then most likely you are looking for the homozygous regions, or if you are looking across the populations or in between populations, of course, you can use other approaches as well. So here I would like to end this presentation. If you're interested in the topic or you want to hear more or something more specific, or even some practical demonstrations of these approaches, be sure to let me know down in the comments. But for now, I thank you for your time and have a very nice day.